Myself, Mr. Mate HT, Department of Botany, RBNB College, Sri Rampur. Today, we are going to study the T.Y.B.Sc Botany, paper first, BO 331, that is the cryptogamic botany. And from cryptogamic botany, topic number 6, that is the life cycle of the Puccinia graminis tritici. Now, from this topic, from this life cycle, we are going to see the sexual reproduction in the Puccinia. Now, it is represented by the two different types of the phases. The first phase in the life cycle is that is the diplophase. Now, this diplophase is occur on the primary host, and that primary host is the monocot host, and its example is wheat. So, all the stages which are under diplophase, they are developed, they are occur on the primary host, that is on the wheat. Now, there is another stage. That stage is called as the haplophase. Now, all the stages which are here on this diplophase, they are occur on the alternate host, and that alternate host is the barberry, and this is the dicot. So, there are the two different types of the hosts which are required to complete the life cycle of the Puccinia graminis tritici, because it is a polymorphic. It shows the 5 different types of the stages. Now, we have to start from the diplophase. So, number 1, that is the diplophase. Now, in this phase, the dicarotic mycelium is getting developed, and it is developed on the healthy wheat crop with the help of another kind of a spore, that is the Acidospore. So, we have to study the germination of the Acidospore and the development of the dicarotic mycelium. So, this is the host epidermis. Wheat lip epidermis, that is the upper epidermis. Here there is the stomata. This is the mesophyll tissue. And this is the germinating Acidospore. This is the aprasorium. This is the germinating Acidospore. This is the germ tube. This is aprasorium. This is infection hypha. And this is the vesicle. And this is the dikaryotic mycelium. These are the host mesophyll cells. Now, in the first phase, that is in the diplophase, the Acidospore is germinated on the healthy wheat leaf surface. So this is the germinating Acidospore. It has two nuclei. One is of the plus strain and another is of the minus strain. That means it is the binucleated. And there are the opposite strain nuclei, that is the plus strain and a minus strain nuclei. And when the conditions are favorable, then in that condition, this Acidospore forms a filamentous germ tube. This filamentous germ tube reaches up to the stomata. And when it reaches up to the stomata, it develops a small circular structure. And that circular structure is called as the apresurium. Then all the content from this germinating Acidospore, that means two opposite strain nuclei and cytoplasmic content is get migrated through this germ tube into the apresurium. Then wall formation takes place in between this newly formed apresurium and this germ tube, and there is a formation of a new structure, that is the apresurium. After this apresurium forms a very small hypa, which is called as the infection hypa. It penetrates through the stomata into the host tissue, and the tip of this infection hypa swells, and that swelling is called as the vesicle. Now, all the content from this apresurium, that means cytoplasm and two opposite strain nuclei, migrated into this vesicle. And after this vesicle, which is the binucleated, it divides again and forms a diploid dikaryotic mycelium. So this is all the dikaryotic mycelium is there. In each cell there are the two nuclei, so this phase is called as the diplophase. In this way, very large amount of the dikaryotic mycelium is developed just below the upper epidermis of the primary host, that is the wheat. Now, this is the starting point of the diplophase. Now, after this, very large amount of this dikaryotic mycelia is formed, and then there is the first spore stage, that is the Urethrospore stage. So, we are going toward the first stage, that is the Urethrospore stage. Now, this is all about the germination of the Acidospore and development of the dikaryotic mycelium on the primary monocot host, that is on the wheat. Now, spore stage number 1, that is the Urethrospore. This is the host epidermis, upper epidermis. This is the host mesophyll. This is the stalk, Urethrospore, host upper epidermis, this is the host mesophyll, this is the dikaryotic mycelium, whole this structure is called as the Urethrosaurus, or it is also called as the Uredia. Now, very large amount of the dicarotic mycelium is formed when wheat crop is young, that is up to the 1 or 2 month. In this way, very large amount of the dicarotic mycelium is formed just below the upper epidermis or up to the stomata, below the stomata. Now, these dicarotic mycelium develop some elongated upright tube-like structures. They are called as the stalk. So, these are the stalk. Now, these stalk, they cuts up a small circular or elliptical cell above it, and that cell is called as the Urethospore. Now, whole this structure is called as the Urethosaurus or Uredia. Now, what is there? Now, there is a very large amount of the dicarotic mycelium is there, then a stalk, or it is also called as the Urethophore, and very large number of the Urethospores are there. Combinedly, all this is called as the Urethosaurus or Uredia. Now, the structure of the Urethospore. So, this is the Urethospore. It has a short stalk formed by the hypa, and above this there is a bi-walled, bi-celled, binucleated elliptical cell is there, which is called as the Urethospore. Now, it has two walls. Outer wall is called as the exine. It is thick and it is rough. Inside this there is a delicate, thin. It is called as the entine. And in the Urethospore there are the two opposite strain nuclei. From these, one is of the plus strain and another is of the minus strain. And toward the pore corner at the middle, there are the 4 germ pores. So, these are the germ pore. Here is one from back side and from front side. That means there are the 4 germ pores are found in each Urethospore. Now, the next stage, that is the Telospore stage. Now, this is the host epidermis, upper epidermis. This is the host mesophyll. Before harvesting of the wheat crop, The same dicarotic mycelium, instead of a development of a Urethrospore, it developed another type of a spore, and these spores are called as the Telospore or the Telospore. Now the structure is the same. That means instead of a Uridia, it is called as the Telia, because there are the different types of the spores and they are called as the Telospore or the Telospore. Now very large amount of the secondary dicarotic mycelium is formed just below the upper epidermis, and also very large number of the upright stalk like structures are formed, and they are called as the stalk, and above the stalk there is a formation of a bicelled binucleated cells. These cells are called as the Telospore. Now very large amount of this mycelia, stalk and spores are formed, which exerts a pressure on upper epidermis, and because of this upper epidermis is get ruptured and all the spores are get dispersed above the surface of the lip, and whole of this structure is called as the Telia or Telospore, in which there is a mycelia, dicarotic mycelium is there, then stalk is there, and very large number of the Telospores are there. Now about the structure of the Telospore, it has a short stalk and it has the two cells, and in each cell there are the two nuclei, that is from plus nuclei and minus nuclei, and this is called as the perfect stage in the life cycle of the Puccinia, because after this there is the karyogamy takes place. So this is all about the second stage, spore stage in the life cycle, that is the Telospore stage. Now third, that is the Basidospore stage. Now third stage, that is the Acidospore. Now here the germination of the Telospore takes place and the development of the Acidospore. Now this is the Telospore. It has the two cells, and in each cell there are the two opposite strain nuclei. And here this is the perfect stage in the life cycle of the Puccinia graminis triticale. Here a karyogamy takes place. That means this plus and a minus strain nuclei in both the cell unite with each other to form a diploid nucleus. Now here in each cell there is a single diploid nucleus. Now meanwhile the upper cell from apex and lower cell just below the septa forms a filament like structure. And these filament-like structure are called as the epibasidia. So in this way, there are the two epibasidia are get developed, and the cell of the Telospore is called as the hypobasidia. So this is hypobasidia, this is hypobasidia, and this is newly formed filament-like structure is called as the epibasidia, and this is also the epibasidia. Meanwhile, each single diploid nucleus from each cell is get migrated into the newly formed epibasidia. So this is the migrated nucleus which is the diploid here. Now the meiosis of this nucleus takes place, and in between all of these four newly formed cell, wall formation takes place. So this is the wall formation, and in this way in each epibasidium there is a formation of the four cells. Now each cell at its apex develops a small tube-like structure which is called as the sterigmata. So these are the sterigmata. And these sterigmata at their apex again develops a small circular structure, and each respective nucleus from each cell is get migrated into the newly formed circular structure, and then wall formation takes place, and there is a formation of the four Acidospores, and from these plus 2 are of the plus strain and 2 are of the minus strain. In this way, from each Telospore, 8 Acidospores are formed and they are haploid. And after this, when they germinate on the next host, that is on the secondary host, they develop the monokaryophase or the haplophase.